అది వస్తే తక్షణ ఈ పేజ్ నాన్ వెజ్ పేజ్ డిస్ప్లే
You mute the background.
Start, sir.
Anup. Okay. So this is a session on the protocol for clinical research. I think this comes in the unit 4 of your clinical research and the pharmacovigilance subject. This is very much helpful for you as well, because for your M.Pharm thesis, you will be you will be preparing the protocol. But one slight difference, the protocol for you M.Pharm pharmacology to this is more oriented towards the clinical research. That's the projects or protocol that prepares for preparing or doing the project in the patients, the clinically oriented research projects. But however, the few of the components from this you can always include in your protocol preparation. So we'll talk about the many factors, like it's a very important document, protocol, for any of research project, because that's the main document as a planned work. The work has to be planned before starting of any project. So this protocol always gives in systematic planning of the work that has to be done during the clinical research. These are all the few things that will be included in this chapter: the definition, clinical research and clinical trial, the clinical research protocol, why protocol, parts of protocol, formats, and clinical research, the Ayurveda, so discussion, conclusion, and also the references. So this is a very basic thing. We call it as research question. Before going into any kind of the research, every one of you as well should have a research question. It could be any kind of project. It could be a clinical project or a non-clinical project, or any kind of project. There should be a research question. Are you all hearing me?
Yes, sir.
Okay. So even for your projects, the research question should be there always. So why this research question is because prior to any research or anything that is there, you should define some problem there, or some undiscovered or unexplored area which every one of us doesn't know. So that is the main thing. So the research is a systematic collection, analysis, and interpretation of data. And also you can see that a research always should be an answer to a question, or there are some problems. So your research should be a solution to a problem. So that's the main thing. Okay, so if you say like you don't know the activity of some drug, okay, and that's the question: what is the activity of this drug on skeletal muscle? If you say example like pharmacology projects, and you don't know whether it contracts, relax, or any kind of causes any other adverse effects or not, don't know. So then you have to conduct a research on that, and you have to find a solution, give an answer to it. Okay, the activity of this drug on skeletal muscle is this. So the whole research component should come here. So always it's not that I will do this or I will do that. So before doing any kind of the research work, you have to identify the what you are going to do there, what it needed to do that, and as I told, like it should be an answer to any question which is not explored before. So we have clinical research and clinical trial. What is there is not much difference between these two, but there is a slight differentiation can be done, the clinical research or the clinical trial. The clinical research is a branch of medical science. It could again systematic, it could be observational and biomedical studies. Whereas clinical trial is a part of clinical research. So research is a very broad terminology, covers the drug from just before lead to molecule, where we have from the chemistry to the drug coming into the market. Whereas clinical trial is a part of a clinical research where you do preclinical trial. Before that, you do all screening of the molecules, and also you do the clinical trial and post-marketing studies. These are all comes, it's a part of the research. We call it as clinical trial. This is also a systematic, but many times this is an experimental biomedical studies. So that's the one difference. And here the ultimate research is ultimate goal is to improve the quality of life many times. And here we specifically see the effectiveness and safety of medications or medical device or any kind of the biologics. So this is what the clinical research is, a very old broad way and more of observations and biomedical studies, and trials is very effectiveness we see that. And as I told, this is how the stages of the clinical trials is classified. Preclinical, that's and exact where the studies goes on in the animals. Okay, and they check the safety, toxicity, pharmacodynamic and pharmacokinetic information here. So once this preclinical trial is done and you get the data, till here there is no regulatory bodies role. But once the preclinical studies are done, you have to submit a form called IND. You heard about it? Investigational new drug, or it also known as investigational new drug application, INDA. So we have to submit this INDA at this level to the regulatory authority, and they verify all the components, and if it is they get satisfied with the reports, only then they approve the drug to come into the clinical phase. So in the clinical phase, we have phase 1, phase 2 and phase 3. There is small group, phase 1. It is done in healthy volunteers only. Phase 1 is not done in patients, only in healthy volunteers. They check pharmacodynamic, pharmacokinetic parameters. And phase 2 is mainly, that is the first in patient exposure. Again, they see the efficacy, safety parameters and kinetic parameters. Phase 3 is mainly of comparative study. They see more of effective thing. These two, or till here, more of safety, pharmacokinetic parameters evaluation is done much. And here, part of phase 2 and phase 3, the efficacy parameters given more weightage here. And they also use the other drug which is already in the market and compare here. So phase 3 is more of comparative studies. So once this phase 3 studies is done, again, the pharmaceutical company, or we call it as MHA, Marketing Authorization MA holder, MAH, he has to submit the another document here to the regulatory authority, that is known as NDA, that is new drug application. So once completion of phase 3, the new drug application is submitted to the regulatory authority, and they verify again, same how they do in the IND here. They also check everything. If it is a satisfactory, then the molecule come to the market, approved as an approved molecule. And even if it is coming after coming to the market, also they do the post-marketing studies. That is known as the phase 4. And from here, every year, okay, so there are we have different timelines. We post-marketing studies for different countries. For first 3 years, some countries has every 6 months you have to submit the report. Some countries are having every 1 year to submit the report. So there are different sequence of timelines in different countries. So at least it will be like 7 to 8 years timeline. The has to be very closely, vigilantly observe what's happening with the drug and report to the regulatory authority. So coming to the clinical research protocol, okay. So it's a definition itself is says like it's a written description. So that's what there is a word in our thing. Whatever you talk, that does not have any much uh, weightage, because people talk until unless it is recorded somewhere. So that's what anything that is written on a paper, it is a document. Okay, so the protocol is a very important document with the written prescriptions, also with scientific rationale. This is very important in any clinical research activity, the rationality. Okay, so because one has to be kept in mind in research, you should not worry about the outcome, whether the outcome comes whether positive or negative, but very strongly they check for the rationality, the need for the study, the research question. So that's the very important component in making any research protocol. Because if you say like you apply for many grants, universities for the protocol you prepare, and you don't know what's going to happen, you don't know the outcome of it. But at that stage, okay, someone should feel that okay, we need this kind of studies. There is an requirement which may bring changes, okay, in the pattern of existing practice in the society. And another thing, they should be helpful to someone, okay, in the society. So you are taking a new molecule, bringing a new molecule, so it will be helpful for patients who have diseases, okay, and get beneficial effect of this drug. Or you're getting a new procedure, then the consultants, physicians, surgeons, they will be helpful. Some devices and some formulations, okay, it helps for the consultants or physician to use. Okay, so you can say that there are only injectables, but you are bringing the oral formulations, okay, then it will be helpful for easy to prescribe and for all the patients. So like this, the rationality is very very important in any kind of the protocol, and what's going to improve in the existing practice. So what's the protocol? If you say that what is main the talk about what is protocol. So it's nothing but it's to clarify the research question, as I told, and to compile existing knowledge. So when you preparing the protocol, like we have to check so much of articles, do the literature survey, check so much of information, and you give a lot of reference. So it gives a very important the knowledge, what the practice that's happening currently. Okay, so that's what it talks about on the protocol, and also to formulate hypotheses and objectives. Can anyone tell like what is hypothesis?
Interpretation.
Hmm? What is
Interpretation.
hypothesis?
An idea.
Assumption.
Okay, idea, assumption. Okay, any other terminologies? Hmm? So you can say that the idea, assumptions, I think both the word will very closely relate to this hypothesis, which is very much, it's not the reality. Definitely, it cannot be the reality. But when you say it is very near to the reality, okay. So one simple example I will tell you how to formulate an hypothesis. If you say like, what is how much it may take for traveling from Bangalore to Mysore, okay. So if you want to know about that, you can see that how you know about that, how you come to know. Hmm?
By calculating kilometers.
Okay, by calculating the kilometers is one thing. Okay, 140 kilometers will be there. Okay, I'll just tell you if it is 140 kilometers, if it is an fully like a where hill stations, how much it may take for driving. Hmm? So you cannot go more than 30 or 40 kilometers in a hill station, right?
Yes,
Not
sir.
even 30 kilometers you can drive. Okay, it's almost takes about 4.5 hours. Whereas if it is a very flat road, very long drive, like we have the road here from Tumkur, this Davangere, which road, it's a very flat road. Okay, 140 kilometers, not even 1.5 hour we can assure travel. And also again the type of road, and okay, there are a lot of things that is there. So now you are building an hypothesis that okay, based on kilometers, you may take so much. And again, you see that how the road it is, whether a single road, double road, hill station, or a flat road. Okay, and by kilometer is one thing which you can say. That another one is like asking someone who has already gone there. Okay, that's the one. So here, what you're saying is this is what you're building an hypothesis. Okay, so they can go like this. They are using this vehicle, and how can I go? Okay, I am using this vehicle and this way. That. So based on the existing prior information, you formulate a hypothesis. Okay, same way the drug. If you say that you have a drug which on the many systems it may act. And earlier studies have shown that, okay, it's an antihypertensive, it acts on the blood vessel, but it also has an activity on thyroid gland. Okay, and their drug is going into thyroid gland. That's what you have, that's what the few information you have. So based on that, you can drive a hypothesis that, okay, this drug is reaching the thyroid gland, whether this drug has an affinity towards any receptors or any enzymes or any hormones in the thyroid gland. Okay, so from there, the existing prior existing information, you are making an hypothesis. You're getting it? So that's the very important. You should have link or develop the existing knowledge to an hypothesis. You cannot say that directly that, okay, I will do this. Everyone can say, okay, there is an one drug, okay, and I will formulate this drug for meningitis. If I say, and a drug is there, considered as an antibiotic, I will formulate this as a meningitis, you are telling. But what is the hypothesis? Okay, so there you should have a strong thing. The drug should be effective against the organism causing the meningitis, and very importantly, the drug should be able to pass the blood-brain barrier. Okay, so all these things should be helpful for you to develop the hypothesis. And above this, and on the objectives, based on this only, the objectives also can be formulated. The hypothesis and objectives are very important component of your protocols. It tells you where you are going, where you are driving. Your research drive is totally talks about in the hypothesis and objective. And to also decide about a study designs. Can you tell what are the different study designs? What are the different study designs? Hello?
So,
What are different study designs? Huh? No one is there, huh?
they're there, sir.
Okay. What are different study designs? Hmm?
Different study designs.
You heard about randomized controlled study? Huh?
Yes, sir.
No. You heard about prospective study, retrospective study, observational, cohort
No,
study,
sir.
case control studies. You have to know this. When is your protocol preparation? Been extended, no?
Yes, sir, it's extended.
But you know about this. It's a very important thing. You should be knowing by this time, because 31st March was last date. You would have been. What are the different study designs are used? Randomized control studies. Very important. If, the subject is assigned to the different groups. You keep it as case group and control group, where one intervention is done in the case group, control without interventions you keep, and check what is the difference between two groups. Okay, so there are so many things which talks about on the study design. You have to go to that, and protocol will explain that. And to clarify ethical consideration, this because your all protocols has to come before the ethical committee. They will definitely see lot of ethical issues. This is one of the very important milestone in any preparation protocol, getting the ethics committee approval. There will be experts in the committee and they see. The ethics is nothing but you should not harm anyone because of your study. Okay. So there was one time there in the World War II, there were people used to caught as a prisoners as a war during the war. The opposite country army people they used to capture as they and keep it in the prison. During that time, they used to do and research on them as a like how you catch the animal and put in the cage and do the research. But later on, it came like it should not never be done unless until the person is voluntarily willing to involve in the clinical research. It should not be done. But unlikely for animals, it is there is no that kind of leniency. But even in the animals also there is an ethical. It should not be more cruelty involved in any kind of research. And also the protocols give for apply for funding and have a guideline and tool for the research team. So this all the major advantages of the protocol. And you can see this the study protocol, how it will be there, like it's a roadmap for investigative team. As I told, it's a plan, show systematic plan, what all should be done at different stages. Also, it's required for scientific and ethical review. For any board you want to apply, like I want to do a research, it's not that just write it in a paper or you just do it in a PPT or you telling orally and say that I want to do this. No, it should be in a protocol. So protocol is a systematic way, as I told, and it's a basis for preparing study reports. Okay, a lot of transparency should be involved here. So these protocols overall will use is clinical trialist. It's also required for the participants who are involved in the study. They should know what you are going to do. The study protocol will give information for the participants. The study protocol will also give information to the funders who give many times I think like our students also applying Rajiv Gandhi University grants, staff also is applying grants. So they do the protocol and send to them. And that's how the funders also consider a valid document protocol as an institutional review board, which definitely is a the thing as I told. Then other people who this is healthcare professionals uh, and also policy makers which uses this especially, and also this is used by the journals study protocols. Now we have a lot of the journals which considers the study protocol, and you can also register in a lot of there, okay, with the journal. So a very good investigator, uh, what he does is the study protocol only they will publish as an article in the journals. Okay, so there should be a lot of information there. So that only you can publish. As we know, like we usually publish some research work, outcome research work, or we publish some review articles, review research work. But here in the journals, they they can publish the study protocol also, even before you're going to do anything. After approval from the IRB, you can publish. So there is an option. This is a very important. Many of us have not explored this place. And also systematic reviewers. It's like information for meta-analysis. What is meta-analysis? Meta-analysis?
The type of study design.
It's also a study design, but what actually how you do the meta-analysis?
By combining data from different research studies.
Yes, that's what exactly. Which already you have done the real clinical research studies, and you compile with the same similar kind of research studies. If you say some other, you can take 5 to 10 other research work who have worked on the similar one, and you are writing an review time. So that's a meta-analysis. So it's also having a lot of great ways, and even the study protocol you can also use for as an meta-analysis. Now coming to the parts of the protocol, this is how, especially for clinical research and typical protocol headings looks like. There should be an introduction to the topic and abstract. What is the overall abstract of the study? The objectives, including the study scheme, the background. It's again little bit of introduction type, but here specifically you talk about on the subject and the rationale, as I told, the rationality, or also we can say that it's a need for the study, and also the eligibility criteria. This is another important, especially in the clinical trials where we do the uh, research work in the patients. The eligibility criteria is one of the important things. And also study design methodology, including drug device info. And what are the criteria for evaluation? So what you are evaluating during the study, especially if you are checking for blood pressure, heart rate, uh, the respiratory rates, and what are the evaluations and what are the criteria that is used, how you evaluate. So that's the thing. And study treatments. So this is the design will come here, especially study treatments, whether it's a single arm, double arm, single blind, double blind, triple blind. So different study treatments how you assign. And the clinical assessment here is what you look out of the clinical factors. Okay, so you look after liver enzymes, you look after the renal function. Okay, how you assess the clinical assessment. And if you say like if you are saying that you are checking any antihypertensive drug to reduce blood pressure, here you have to say that okay, drug reduces some 5 mmHg of blood pressure. Then if you can you say it is effective or not? So how much it should reduce? So all these things it should define here in the clinical assessment section. And also clinical laboratory measurements, different tests that has to be done, and also evaluations by visit like adverse drug reaction reporting and documentation, all those things, and also the discontinuation. So on what particular criteria you discontinue the drug. Okay, so and what you would do for after discontinuation. So there is an ethical issues comes here always. So what happens to the person? So in the animals, okay, you have somehow you can manage, but in the human beings, so sensitive thing, something happens to human being, so you cannot leave him in between, okay, because your drug is discontinued. And for those patients, how you manage if something happens. So all those plans should be there, and how you replacement of subjects, other people, how you bring protocol violations. Um, See, there are so many rules and regulations in our routine life. It's very difficult to follow everything if you say practically. So, same like in the clinical research field, also very difficult to follow all. So, definitely there could be some kind of violations that can occur here and there. And here you should mention what are the possibility violations that can occur, and also give a solution that how you can manage that violations. or what are the alternative routes to rectify any errors because of the violations. So that also should be there. And also statistical section like analysis and monitoring. And also administrative, ethical, regulatory considerations. Reference and appendices always should be there. Also publication part also should be there. And what's your main, how you're going to publish this article, and in which planned journal, and what are the different timelines you are publishing during the study. So all of this information should be there in the protocol. So one thing is, in your protocol all these components may not be required, but few of these components are definitely will be there in your thing. But mainly I find when a pharmaceutical company is doing a clinical trial or not, the company a very systematic trial, all these components will be certainly part of the protocol. So coming to the introduction, as I told, like study subject, you should talk about if you're talking on doing a research on like any hypertension. Okay, so you have to talk about what is hypertension and how it usually, what are the risk factors, what are its clinical manifestations, what are the complications, what are the different types, pathophysiology, and also you should talk about antihypertensive. What is the problem with the antihypertensives? What are the drugs used? And how the people are suffering from hypertension? What is its economic impact? All those things you can include. Exactly. So you should talk about the study subject and the hypothesis. So hypothesis also you should bring. At the end of introduction, you should bring that, okay, all this problem are there. That's why I'm doing this study. So should be clearly identified and briefly described in introduction. So that's the important thing. And in the introduction, you should never go out of your uh, the topic of the research. Okay, if it is unnecessarily, you should never include any other information. Okay, so if you're saying that you're talking about, just give an example, you're doing an hypertensive study and you are looking for a drug like beta blockers which acts on the beta 1 receptors. Okay, so beta 1 selective receptors you're talking about, and you want to do a research on that and find what all the conformational changes that occurs in the beta 1 receptors. So this is your study. So when you're writing the introduction, you should be more focused on the hypertension and how the beta receptors have the role in the hypertension, and how this antihypertensive beta blocker acts on the receptor. But here, if you include like unnecessarily, like, okay, what is the role of patient counseling in hypertension, or what is the non-pharmacological dash diet, and all those things, no need to include in the introduction at all, even though that is a part of hypertension. But because your the study is mainly focused on which area in the hypertension, so the most of the information that included should be from the, it should revolve around your research topic. Okay, are you all following?
Yes, sir.
So that's a very important thing, which specifically talks about the introduction itself. We should. So here, most of the time, you should also include that what are the problems associated, what is the issues there. Okay, and lastly, in the end of the introduction, you should say that, okay, my research goal is going to help this. So that's how the introduction should be written for any protocol. The next is the abstract itself is the thing. So it should a summary of the information in a document, the overall what is done. So it's usually in very limited characters should be used here. Sometimes 250 or 500 characters. and it's an overall summary. And here in the abstract, none of the figures, tables, all those things should be used. And one more thing I just want to miss in the introduction, So better not to use any kind of tables here in the introduction. So only if it is so much necessary and so specific to your study. Okay. So example, if I say you're looking for an beta blocker on beta 1 receptors action, no need to put hypertension classification table. No need to put dash this one table, this one, and no need to put what all the different table in the complications, all those things. Okay, no need to put the classification of the drugs table. So you should never don't use that. Only if it is something that talks about beta blockers, some table is there. Okay, so only those you can use. The two is a limitations, not more than one table or figure should be used in the introduction. But in the maximum, you have to avoid this introduction, the tables or this one in the introduction. So in the abstract also, it's all more fully, it is 100% only the characters, no tables, figures should be used. You can see this, it should revolve around all these things. So there should be an hypothesis, there should be a materials, what it is involved, there should be a procedure, okay, there should be a statement, and lastly there should be a conclusion out of the thing. What you are going to conclude. So it should be very accurate. As I told you, there is a limitations here. You cannot go more than one page of abstract. You cannot write 5-6 pages. So it should be accurate, reflects the purpose and content. You should go to the directly to the point. No any long sentences or just sentences which gives a very vague information and not specific should not be used in the abstract. So it should be concise and specific to make sentence informative and as possible as brief. And also it should be non-evaluative to report information objectively. And next is the objectives of the study. This is one of the thing like the title. It's totally based on the title. Always here you should correlate very importantly the title of the research, the objective, and your outcome. So title will talk about something like what you're going to do, and to do to that titles, what all you are put in the titles, to make it true or to check whether that has to happen or not, the objectives helps it. So it should revolve around that. So if you're talking about hypertension, beta blockers, beta receptors, okay, your objective should be totally there. Should revolve only with that. So the question, research question, what it is usually is reflected in the title, and what the title is saying to bring about that the objective should be there. So you say like should be clearly mentioned as hypothesis. This is how it comes. What you want to be tested, the hypothesis. And also should have a corresponding discussion in the statistical section. So always when we are evaluating any student's project, myself what usually I do is I keep only 3 things in evaluating any protocols or the thesis. One is I keep the title, I keep the objectives, I keep the results. I usually don't see more about other section. So I see whether what you are asking in the title is you are mentioned in the objectives. and what you have mentioned in the objectives, there is a result. Okay, so only then the whole protocol, the research study, the aim will get fulfilled. Okay, so one of the most common errors the students does is these three things. So they put title somehow one thing, and if you see to the title and the objectives, many times the all the objectives you given 3 objectives or 4 objectives, many times they will not be relating at all. They will be telling some other thing in the objective. You're going out of your the study title. Okay, if it is there something in the objective, ensure that it should also should come in the title. May not be specifically, but in that area something should be covered in the title. And same way, people give some title, give some objectives, and if you see the results, I'll see a lot of what are the objectives, and for that objective, what the result is given in the each section. At the end of the, I'm telling about the evaluating of thesis. But each objective should have a result always. Only then study will be fulfilled. So here the objectives can be two things. One is primary objective and secondary objectives. The primary objectives always will be one. So number of primary objectives most of the time will be one primary objectives, or sometimes the even two can be added, not more than two. Very rarely we keep it as two as primary objectives if it is definitely required. There is one primary objectives will be always better, and the primary objective should be always a very broad terminology. You don't keep it as very specific, and this primary objective should always like how you keep the title where it covers all the components of a research. Same way the primary objective also should be there. Next is the secondary objective. The secondary objectives can be many, and it should not be one, it should be multiple. So it's almost opposite to that of primary objective. Primary objective always one, secondary objective many. So this secondary objective should be very specific. Okay, and specific in sense like it should be totally different than the other objective. You cannot do like I am saying that I am hypertensive. If you say that secondary objective, it could be okay. Primary objective I can keep for the hypertension, like to investigate the conformational changes of beta 1 receptors to beta 1 blocking drugs. Okay, so maybe a selective beta 1 blocking like an atenolol. Okay, like that I can keep the drug as a primary objective. It's a vague and same like not much specific. Secondary objective you can see tell that like one objective it could be to identify the genetic variations. Okay, means different genetic variations like different people, blacks, white, is there is any changes in the beta 1 receptors and beta blocker activity. The another secondary objective you can tell that what is the you are finding like the the duration of beta 1 blocking on beta 1 receptor. Okay, so that is as one objective. The another objective you can put it as like what is the after the effect, what are the secondary effects because of beta 1 blocker on the beta 1 receptors. Okay, and the another objective you can keep it as a lot of other the structure activity relationship SAR of beta 1 blockers and beta 1 receptors. So like this, there should be secondary objective. You should mention very cleanly the specific items that's going to do, and one secondary objective should not come in the other one, it should not overlap. And secondary objective, as I told, should be more, not only one. You can keep it 4, 5, 6, 7, 10, no problem. But for every secondary objective, you should have 1 or 2 results in the different way in the result section. Okay, so that you have to ensure that every objective will have a result in the result section. Are you following?
Yes, sir.
So the objective of the study is very important in this sense. Next is the background. This is also more of like an introduction. But the introduction you talk about, give some information in and around of your topic. Introduction. But background you will give very specifically, like detailing the rationality, scientific rationality. Why you want to do it, what are the existing issues and all those things, and the justification. So rationality, justification, so in using any medical and scientific literature. So what the title of the research question, what the that is also known as hypothesis you proposed, you should have a very strong evidence to tell that there is a problem here. So that's the thing which you usually give it in the background. But many times the introduction or the background, usually some journals they give introduction, some they give it as background. Most of the time it is used synonymously, or many time information is written same thing in the introduction or the background. But there is a slight difference. As I told, introduction you'll talk about in and around of the topic, and background you see specifically your hypothesis, what the thesis of your title, that, and you have to give the rationality, justification, and see why this study has to be done. So that's the information in the background. And also study rational, this also comes make same thing, like why it makes sense to study this product in this patient population, or any observation study, why the information is needed, risk benefit assessment. So all these things will be elevated in the study, and why the potential benefits outside the risk, and should be clearly mentioned about this. So it's almost background, study rational, it's also same almost. The next is the eligibility criteria. So eligibility criteria in any clinical trial is one of the very important aspects. So you can see that you see like what kind of people you will be able to study in this study. If I continue the same hypertensive study, okay, you are using the beta 1 blockers to study on the beta blockers, beta 1 receptor. But sometimes these beta blockers will also block the beta 2 receptors drugs. And if you block the beta 2 receptors, which are specifically in the lungs, so those people will get constricted because beta 2 receptors is a vasodilator. Okay, so it should not be used in people like having asthma, COPD, or any pulmonary fibrosis, or any kind of the pulmonary cancers, lung cancers. This kind of things you should never be used. because it further deteriorate the patient's conditions. So during that time, you should tell that patients having this disease are not included in our study. It is excluded. So like that, we should have inclusion criteria and exclusion criteria. Which are all the patients which can be included in our study and who should never be included in our study. And also we have to give the reasons for why you are including some kind of patients and why not. So if I say like again, if I say that okay, patient is an hypertensive, okay, from last 5 years, only then I will include in the study. Okay, why you are including only patients who have hypertension last 5 years? Then you should have a rationale. Yes, because I am studying the effect of beta blockers on beta 1 receptor. So if the patient is taking some beta blockers or any other antihypertensives and patient is hypertensive since last 5 years only, the beta receptor will be active again so that I can do a study. But more than 5 years patients, there is a down regulation of beta receptors. Okay, so if there is an information like that, then if there is a down regulation, like okay, down regulation means no, what is down regulation and up regulation? So down regulation in sense like if there is a decrease in number of receptors, then you will not have the able to study the beta blockers. So that's how the research this one it goes off. Okay, and that's why the reasons rationality for making not to uh, include the patients within the last 5 years. So that's what the reasons imposing eligibility criteria. You can include rationales, safety concerns, regulatory issues, and the practical considerations. So these are all the few things which usually helps in the formation of the eligibility criteria. Okay, you're all there?
Yes, sir.
Yes, sir.
So we'll stop. We are